and if it's possible to refer them to the tissue. And if it's possible, after that, identify what type cells in this tissue on the electron micrograph. So let's look. This is one or more than one cell. This part of one cell. We can't see cell membrane at all. What we can see? So this part of one cell. So after that we can look at the organelles that help us to identify what is a tissue cell. We can see nuclear, because double membrane. This is mitochondria, also double membrane and crystal inside. And what is it? This is myofibrils, striated. So this is striated muscle tissue cell. That's all. Not so. Also, we remember that muscle tissue we subdivide into skeletal, cardiac and smooth. Because it's striated, it may be skeletal or cardiac. Have we some information to determine the skeletal or cardiac? Yes, why? Because position of nuclear in the center between. So this cardiac. That's all.
Teacher, can you identify here the bands, A band and I band?
Yeah, if teacher asks you, you should identify Z line, this A band, I band.
Okay, again, teacher, this two is the
Z, this one sarcomere. Each sarcomere consists of one A band and two half of I band. And what this is H band and in the middle M line. So this very thick and thin filament cross, this dark enough. This very portion, very thin filament finish will be a bit lighter. This A corresponds to H band. And all thick filaments arrange with each other this M line by myomycin protein. So, no question about this? Next. This is how many cells here? One, part of one cell. Okay, let's look what we can identify. This is double membrane, this part of the nucleus. Look, no heterochromatin. So, very active cells. And do you remember what the most active cell in our organism that have light nuclei? Neurons and hepatocytes. Let's continue. What we can see? We can see, yes, complex, rough endoplasmic reticulum with free ribosome. This is nasal body. We can see a lot of cytoskeleton elements, mitochondria. So this is body of neuron. Neuron. Have you any question? Maybe something. Golgi apparatus visible here. Microtubules, single line neurofilaments.
This is microtubule and this is neurofilaments.
Microtubules to parallel line. What's these microtubules to this, and single line is neurofilament. All them we refer to cytoskeletal system.
This one is nasal body.
Nasal, all these particles, because nasal consists of complex free ribosome and riboplasmic reticulum. Later. Okay, what can we see here? No. Yes, you're right. This, look please, what we can see, very thick basement membrane. Such thick basement membrane we can see only glomerulus of kidney. Okay? So, look please, this is lumen of blood vessel. And blood vessel, what type of blood vessel? We can see fenestra, fenestrated blood vessel. In the lumen we can see what? Erythrocyte, this nucleus of endothelial cell. And basement membrane is general as for the blood vessel as for the podocyte. That's why it's very thick. And these peduncles of podocytes. So this barrier. Do you have a question? No? Again. This nucleus of endothelial cell.
So the middle there is another
Cell.
This is part of podocyte. podocyte form peduncle on the basement membrane. This part of one cell, and we can see myofibrils. Z line, I band, A band, mitochondria. So this striated muscle tissue. What we can identify because presence of secretory granules. It's cardiac because in atrium there are special secretory cardiomyocytes that produce what hormone? Antidiuretic. And this smooth endoplasmic reticulum, it's well developed in striated muscle tissue, and this is glycogen inclusion. What is it? Look, we can see one cell, very clear cell membrane. This cell have basement membrane or not? No. So this cell belong to connective tissue. Now let's look at the organelles. We can see nuclear and large granules inside. Yes, it's mast cell. Do you remember what substances mast cell produced? Okay. Next. Now we can see junction here and myofibril. So this is striated muscle tissue and cardiac because we can see intercellular intercell junction, intercalated disc. Yes. Do you remember what part of intercalated disc consists of? Gap junction, transverse portion and lateral. Where is transverse? We can see desmosome. Where is lateral? Will be gap junction. Gap junction will be between myofibrils, parallel myofibrils. Transverse instead of the Z line. But here must be Z line. This will be transverse portion. And thin filaments joined to the membrane. This is thick filaments, again thin, Z line visible here. H band, M line in the middle. This is mitochondria. That's all. Next, let's look again. Again, you can see my striated muscle tissue, but because we can see secretory granules, this cardiac, yes. And this, look please, this is membrane, sarcolemma, together with basement membrane, space intercellular, and another cell. We can see, I think, basement membrane here and a lot of vesicle. Maybe this wall of the blood vessel, transport vesicle. Okay, how many cells here? One, part of one, yes. We can see nuclear, a lot of rough endoplasmic reticulum. So, and look please, what this we can see cell membrane that underlying basement membrane. So, what tissue? Epithelium, yes. Can't be connective, can't be muscle, not nerve, epithelium. And because of a lot of rough endoplasmic reticulum, this is what type of epithelium?
Glandular epithelium.
We can't see, unfortunately, apical surface where it will be storage of secretory granules. We can see only basal, but because we can see a lot of rough endoplasmic reticulum, we can say that function of the cell secretion, so this glandular epithelium. Yeah? And tell me please, is mucus or serous cell? Serous, rough protein, yes?
Ma'am, what's this?
This mitochondria, not good, small magnification, not good section, oblique, so it's difficult. No, this mitochondria. Okay, what is it? We can't see cell membrane, this part of one cell, yes? Let's look at the organelles. Rough endoplasmic reticulum a lot, mitochondria, secretory granules. So probably this epithelial cell, glandular epithelium again. And look please, we can see around one granules a lot of membrane. Actually it's maybe Golgi apparatus surround these granules. These are major, these more darker than these, yes? These are major granules that are going to leave Golgi apparatus subsection. This is what? Nucleus. Yeah. Again, part of one cell, and we can see what? Rough endoplasmic striation. Yes, myofibrils. And they range, we can see sarcomere. It's oblique section. Now, what this very thick and thin filament, this that line. So, because they maybe go in different direction, and we can see secretory granules, this cardiac. This is Golgi apparatus. This
Mitochondria.
okay. This part of one cell, and we can see what organelles? Mitochondria. Because we can see double membrane, and look at the crystal, at the shape of the crystal. This tubular vesicle type of crystal. Such mitochondria exist only in such cells that produce lipid. And we can find such mitochondria in the cortex of adrenal gland. Steroid hormones. And between mitochondria, a lot of smooth endoplasmic reticulum, lipid droplet. They use it for the synthesis. Cells use. What is it? Maybe nucleus. Lipid, yes. Lipid droplet. Again, part of one cell. And what we can see? We can see free ribosome and rough endoplasmic reticulum. So complex again. So this is nice body. Golgi apparatus. So what is in tissue? Nervous. This body, nice body, can be found or in body cell or in dendrites.
This is what teacher.
These secretory granules, we know that they secrete something, so this may be acetylcholine. This is destroyed mitochondria. They very sense what this is and this mitochondria. It's sensitive for changing pH, and during preparation they can be destroyed. This, look please, what is it? Straight muscle tissue, and this invagination T-tubules. What all these electron micrographs demonstrate for you T-tubules of striated muscle tissue. Look please, what these T-tubules opposite that line, so these electron micrographs correspond to the cardiac. Here one T-tubules, one more, but we can't see opposite what structure. That's why we can't say exactly. This may be skeletal, maybe cardiac. And these, we can see myofibrils, that line, and this system of smooth endoplasmic reticulum around the arrangement of smooth endoplasmic reticulum around the myofibrils. What function of smooth endoplasmic reticulum? Storage of calcium. Calcium necessary for initiation of contraction.
Calcium.
Yes, storage of calcium only in muscle tissue. We can see one cell, one more cell. These two cells underlying by basement membrane. These cells situated close to each other. In the cell we can see part with rough endoplasmic, apical portion with secretory granules, so polarity sign. So this epithelial tissue is glandular epithelium. Let's look here. We can see two flattened cells. They joined with each other by intercellular junction. Yes. So this also epithelial cell. They flatten simple squamous epithelium. So what is it? Endothelial cell, lumen of blood vessel, the basement membrane. Between two types of epithelial cells, we can see process of cell without basement membrane. So this process can belong to connective
Connective
tissue.
tissue.
Fibroblasts, they have process, yes? Nervous process. Значит, this is cross section through the myelinated nerve fiber and unmyelinated nerve fiber. Between them we can see collagen fibers. Yeah. Here axon, external mesaxon, internal not visible. No, it does not matter. This we can see a lot of axon inside of Schwann cell. This is nucleus of Schwann cell. This is cytoplasm of Schwann cell. Yes, these myelinated sheets which consist of the double membrane of Schwann cell.
This is what tissue?
Axon, nervous
This is
process, nucleus of Schwann cell. This is again this myelinated nerve fiber. Inside will be axon. No, that's all.
Myelinated.
Myelinated. What? Myelin sheets. This
Unmyelinated.
unmyelinated. This unmyelinated. This myelinated. But this oblique section, yes, these collagen fibers. Okay, to cell, because we can see myofibrils and junction. So this cardiac muscle tissue, mitochondria, and this look please transverse portion of intercalated disc which consists of desmosome, and this lateral portion where is two membrane will be close to each other, nexus. Так, господи. Значит, let's begin. This is longitudinal section through the nerve fiber. Myelinated, but we can see myelinated sheets. This region of node of Ranvier, yes. This is what this portion, high magnification, with nerve fiber, this cytoplasm of Schwann cell, basement membrane, upper. What is it? This is central nervous system, and we can see a lot of nerve processes with mediator, with transmitter inside that are going to form synapses on each other. Some of them transparent, some with dense core. Some, for example, acetylcholine, they transparent, and these may be neuropeptides. Because this we can see mitochondria, Golgi apparatus. What is it? Centriole. So I can guess that something here will be borderline of nervous cell. Nervous cell, because organelles belong to the nerve. This is processes that come to this nervous cell are going to form synapse on the body. But if you remember, we name this as neuropil, because sometimes it's understandable what process come to which, where synapse, and so what all this we name as neuropil.
This one is collagen also.
Collagen, yes, collagen outside. Значит, this we can see junction between this basement membrane and what is it? And this muscle cross section through the what band? This what band? A, because thick and thin visible, very clear. This where is oblique, where is only thin filament, this that line. Because we can see junction between two adjacent, so this cardiac muscle tissue. Let's look here. I think this also cardiac muscle. Let's look at this. We can't see any basement membrane, so this part of cell belongs to connective tissue. We can see rough endoplasmic reticulum. This is fibroblast. Let's look at this. We can see axon inside of the cytoplasm of Schwann cell. This is unmyelinated nerve fiber that going to innervate. Так. Again, This is, we can see mitochondria with tubular-vesicle type of crystal. This may be Golgi apparatus, may be smooth endoplasmic reticulum, it's unclear, but we know that such type mitochondria we can see in the cells that produce steroid hormones. What is it? I think these electron micrographs demonstrate for you Golgi apparatus. Golgi apparatus destroyed mitochondria and other structure understandable. And you should remember function of Golgi structure and function Golgi apparatus. So cis phase, trans phase, three portion flattened cistern that have curvature and some small vacuoles around and large vacuoles. Main function of Golgi apparatus, production of primary lysosome. After that, one more function, proceeding, the modification and transport outside of the cell, and one more, take part in renewing of cell membrane.
Oh, yes.
Next, this we can see part of one cell, this we can see cell membrane, and this cell cover another cell. After that will be basement membrane. Nuclear, very light. So let's look at the cytoplasm. Small Niesel bodies, very low magnification. These neurons, and neurons covered by satellite cell. This again neuroglial relationship. This is neuron, nuclear of neurocyte, this very small Niesel, small particle of Niesel, and this we can see glial cell, satellite cell, and only after that we can see basement membrane. Okay. This and this. This is muscle tissue, striated muscle. We can see arrangement. Here we can see T-tubules, and if it's opposite Z lines, this will be cardiac. But we can't determine, no details, no information. Here, blood vessel, in the lumen we can see erythrocyte, this nucleus of endothelial cell, this pinocytotic vesicle transport process, collagen fibers. These electron micrographs demonstrate for you terminal of nerve process where we can find mediate transmitter, transparent, and with dense core transmitter. So this nervous system, this nervous process, we can't see formation of classic synapse, maybe what here will be later. Only storage of mediator. Maybe on the next section we can see structure of synapse. So what, for example, terminal and form synapse on the body. And if we cut through here, we can see structure of synapse, but we cut it a bit upper. That's why we can't see presynaptic membrane, synaptic cleft. We can see only process of with mediator, with transmitter. Is it clear? Other, understand, what this, I can say these collagen fibers outside of basement membrane. Other, neuropil. What is the tissue? A lot of cell we can see, and this, look please at this. What is the tissue all this cell belong? Muscle cell, yes, striated muscle tissue. That line, this oblique section, A band, I band, this is cross section through the A band, thick and thin filamen, this only I. That line. This skeletal or cardiac? Look attentively. What is it?
Junction.
Junction. This cardiac. Now, so this one cardiomyocyte, one more. What is it? Between adjacent, we can see blood vessel. Blood vessel with lumen. And look at this. What this structure and this structure. What is it? Between cardiomyocyte, we can see again unmyelinated nerve fiber. Axon inside. Axon inside of Schwann cell. And this very thin cell without basement membrane, fibroblasts. And one more electron micrographs. It's dark enough. Look please, we can see rough endoplasmic reticulum, Golgi. It's underlying by basement membrane, so this epithelium. And this we can see lumen. So yeah, this blood vessel also underlying by basement membrane. That's all. Again, we can see, what now we can see synapse. So look please, what this junction, this presynaptic membrane, why? Because here we can see mediator vesicle, this postsynaptic membrane because vesicle absent here. Mitochondria, so this again neuropil, and we can see synapse structure. This is again synapse structure, what junction? Presynaptic membrane will be left or right? Left, because we can see the vesicle. This also nervous process with mediator. This one more synapse. Так, this and this synapse. Let's look again at this and this. This again demonstrate for us different type of mitochondria. This classic mitochondria with flattened type of crystal, this tubular vesicle type of crystal, and we can see this lipid droplet, this adrenal cortex, this any cell. This specialization of cell membrane, we can see cilia, this cross-section through the axonema, it consists of the 9 duplets plus 2 in the center. This basal body through the base, it's equal structure to the centriole, 9 triplets. This longitudinal section, we can see axonema through each cross-section, and this basal body, through which what this corresponds. That's all. This we can see 1, 2, 3 cell. These 2 cells without basement membrane, so they belong to connective tissue. This is muscle tissue. We can see myofibrils striated, yes. If this is invagination correspond to the T-tubule, opposite line, cardiac. Let's look at this cell. In this cell we can see a lot of rough endoplasmic reticulum, so probably this fibroblast. There we can see maybe this secretory or lysosome. Sometimes it's unclear. Let's look another organelles. If it's secretory granules, we should find rough endoplasmic reticulum. Rough endoplasmic reticulum absent. Yes, we can see only free ribosome, so cells produce protein for itself, maybe for the primary lysosome. So this probably secondary lysosomes. So this is macrophage. Так, 1, 2, 3, 4, 5, 6. This is muscle. We can see myofibrils. This is sarcomere structure, that line. And what if this invagination beginning formation of tubules, so we can say that this is all cardiac. Let's look at this. This cell without basement membrane, rough endoplasmic reticulum, this fibroblast, we can see collagen fiber around. What is it? Blood vessel, yes, luminal blood vessel and basement membrane visible, very clear, yes? Так, again we can see what?
Synapse.
Synapse, because these mediators, so this presynaptic membrane, synaptic cleft, postsynaptic membrane. This is Nielsen body. These collagen fibers between two adjacent cells. That's all. Uh, this we do the same, but with this new. Look, please, this oblique section through the unmyelinated nerve fiber. We can see axons that surround cytoplasm of Schwann cell. Between them, we can see connective tissue with collagen fibers. And this one part of fibroblast maybe perineural cell. Perineural cell must be surrounded by basement membrane from both sides. Connective tissue cell, no. Let's look attentively. Without basement membrane, so this connective tissue, this one part of nucleus, very small. We can see rough endoplasmic reticulum here, this fibroblast. This part of one cell, and we can see, look please,
This is the body.
Nielsen body, a
Mitochondria.
lot of microtubules. So this may be what? No, this body of rose.
Body.
In body we can see a lot of maybe body, but maybe dendrites. But because in the large dendrites we can see a lot of maybe formation of dendrites near the body, because a lot of microtubules. What is it? Muscle cell. And look please, what this cross section, this oblique, so no very good arrangement. So if you remember, among the cardiac cell, cardiomyocyte, we can find conductive system. Conductive system cell actually arise from typical cardiomyocytes. But there we can find less myofibrils and they not properly arranged. So what these cardiomyocytes that belong conductive system. Centriole, maybe Golgi apparatus, mitochondria. Так, again, muscle cell, striated muscle, yes, так, what is it? This that line or junction? It's unclear. But this actually, if you remember, demonstrating for you secondary lysosome. Secondary lysosome usually consists of different portion, heterogeneous contention, light and dark. Because after fusion primary lysosome and phagosome, will be formation of secondary, will be different part. Так, here also look please, what this is, secondary lysosome, it consists of a lipid droplet I think and something else. All these mitochondria, this is myofibrils, that's why this also in muscle tissue. We can't say what is the cardiac or skeletal. No information. Так, this, look please, this is one cell, this is one more cell. Junction between cell, rough endoplasmic reticulum, secretory granules, this glandular epithelium. They form canal to transport secretion. And this cell without basement membrane, so this cell connective tissue, belong to connective tissue, with such process, segmented nuclei, so this is cell of blood. But some of them in the lumen of blood vessel, when cell leaves the blood vessel, they form pseudopody, form movement. That's why they look like this. So because granules very small, this may be neutrophil. This also very high segmented nuclear neutrophil in the lumen blood vessel. We can see in the endothelial cell. Так, this cell we can see very large granules. It's not nuclear with such process, so because very large granules, this basal. Very good.
Teacher, is eosinophil?
Where is it? In the eosinophil, we can see special type of granules with dark, they elongated and crystalloid in the center. We can't see crystalloid here, so it can't be eosinophil. It's very good. One part of cell with, what is it? Needle body. So this part of neurons, we can see Golgi apparatus, mitochondria here. Very good electron micrographs. This muscle, tubules opposite the line, cardiac. Again, neurons, yes, needle body. And look please, what this is, neuron covered by glial cell. Only after that will be basement membrane and these collagen fibers visible. So this glial cell, this neuron. These some process come to the through the body of one cell or glial cell come to this body to form synapse maybe. This tissue, muscle tissue, junction, so this cardiac. And nuclei approximately in the center, yes, this is blood vessel. But this, if we can see very clear, no, look please, what is it? Oblique section. This is unmyelinated, no fiber. Axon, fibroblast here.
Apa tu?
What is it?
Ini fibroblast ni unmyelinated axon.
What is it? Two cell, nuclear, rough endoplasmic reticulum, glandular epithelium, secretory epithelium. Again. This is tubulovizygal type of crystal, adrenal gland, cortex adrenal gland. Oh, what is it? 3 cells.
Smooth muscle.
Smooth muscle, yes, we can see dense body, a load caveola. Caveola function what? Storage of calcium. Near the nucleus, we can see a lot of organelles, rough endoplasmic reticulum, what is it? Centriole, mitochondria, and all these cytoplasm filled with actin. Myosin also present, but it's not visible. Why? Because it has special structure. Yeah? Do you remember? And process of activation, initiation of contraction connect with changing such shape of myromyosin, no, not heavy, the light myosin, straight, from curvature to straight. This also smooth muscle cell, nuclear, this all actin. Dense body, mitochondria, dense body, and what this is caveola. And this is what? Eosinophil. Why? Because if we look attentively, we can see dark crystalloid inside each granules. Such granules can have only eosinophil, can be found only in eosinophil. That's all.